The next speaker is Dr. Anna Babayan. She is from the Illumina Cancer Research Center in the UK. Dr. Babayan did postdoc in translational cancer research and liquid biopsy at the Hamburg University Medical Center. She has been involved in the Innovative Medicine Initiative Consortium on the Cancer ID project, which aims to generate precision blood-based biomarkers such as circulating tumor cells, cell-free DNA, and microRNA, circulating microRNA. She's currently the Cancer Research Marketing Manager for Illumina in Europe, Middle Eastern, and Africa. Unfortunately, Dr. Babayan cannot be here today due to a recent surgery. We wish her a very prompt recovery. And today she will be providing us a talk on multi-omics technologies in cancer research.
Hello. Can you hear me?
Yes, perfectly.
Thank you. Thank you very much. Thank you very much for this nice introduction. Indeed, unfortunately, I could not go to Spain. 2 weeks ago, I had a surgery, and my doctor recommended me not to fly. Really a pity. I would like to be there with you, but thank you still for giving me this fantastic opportunity to speak at the workshop. I'm really happy to have this chance, and I hope you can see my slides well. So today I would like to speak with you about multi-omics. So just briefly, my name is Anna Babayan, as you already heard, and I'm a marketing manager for cancer research at Illumina. And in today's presentation, I will be discussing several next-generation sequencing technologies for multi-omic approaches, reviewing a few interesting studies that have been published recently and utilizing also Illumina sequencing platforms and workflows that can be used to support similar studies you might be thinking of performing. For those of you who haven't heard of Illumina, we're a global company and we develop, manufacture and market innovative next-generation sequencing and microarray technologies. Our technology enables you to make foundational discoveries, transform medicine and ultimately impact lives. To date, over 220,000 publications involve the use of our technologies, clearly showing that this is a robust technology you can trust. And you can get access to our technology in one of two ways, either in your own laboratory or through one of many service providers that are found throughout Europe. But let's talk science. What are omics? And what information do they add to cancer research? To answer this question, let's take a step back and spend a few minutes talking about the hallmarks of cancer. We know that all cancers acquire universal hallmark characteristics. And despite commonality, cancer hallmarks are acquired in different ways. The introduction and the discovery of hallmarks basically endorsed the era of precision medicine, as different mechanisms by which hallmarks are acquired can be therapeutically exploited. And thus therapeutic options are no longer limited to cytotoxic and untargeted chemotherapeutics. Deeper understanding of cancer biology has led to the paradigm shift from imprecision to precision medicine. On one hand, we have therapeutics targeting specific hallmarks and pathways. And on another hand, precision medicine relies on identification of biomarkers, which are crucial for stratification of patients to select those who will benefit from a given therapy. So the question is, can just one biomarker stratify patients for therapies? The answer is no, unfortunately. So even if therapeutic option is developed to target genes and pathways specifically altered in patients' cancer or cancer subtype, success of the targeted therapy is highly variable. For example, here on the left-hand side, up to 1/3 of triple-negative breast cancer patients have a proven BRCA1 and 2 mutations. And these patients could potentially benefit from PARP inhibitors. But we know that only up to 40% of these patients really benefit from PARP inhibitors. Similarly, about a quarter of breast cancers overexpress HER2, and thus they could potentially benefit from anti-HER2 therapy. But we also know that up to 60% of HER2 enriched tumors do not achieve a complete response under anti-HER2 therapy. And it's even more complex in immune therapy. None of the currently existing biomarkers can reliably predict outcome. And we will speak about it in a few minutes. But the question is really, why are so many patients failing to undergo remission following precision medicine treatment? What we're essentially missing and that very few cancers can be explained by one factor alone. For instance, while some genes predispose an individual to developing a particular cancer, one's genetics are not 100% predictive of disease development and how the disease will progress. So other modalities of cellular regulation, such as epigenomics, transcriptomics, proteomics, regulatory factors, they all may also contribute to cellular transformation and cancer progression. It's also apparent that there are myriad of extracellular factors that also influence cancer initiation and progression. And obviously, it's not only the sum of these factors, but also the interplay of the multiple factors. It's now clear that gaining a more comprehensive understanding of the many factors influencing cancer are crucial to cancer research. So despite the fact that we have determined a patient's genetics and a pathway that should be therapeutically addressable in their particular cancers, we are still clearly missing something. I'm sorry, here we go. So let's talk about multi-omics. As we are going to discuss in today's presentation, the missing puzzle piece issue may be solved in a manner that embraces the inherent complex biology of cancer. Traditionally, next-generation sequencing has been implemented in cancer research to do characterization of one omic per experiment. So, for instance, whole genome sequencing experiment may be completed to understand the mutational profile of cancer, which is incredibly useful. But we know that cellular regulation in cancer is incredibly complex and cannot be explained just by one mutational profile alone. And similarly, if we were to characterize any other omic in isolation, like for example, characterizing cancer cells proteome alone or characterizing epigenome alone, we would still have an incomplete view of how the cancer cell is regulated. And in reality, cancers are influenced by a myriad of factors at once. Here you can see four, let's say, major omics like genomics, transcriptomics, epigenetics, transcriptomics, and proteomics. But there are, of course, more omics regulating cancer initiation and progression. Also, we have metagenome, secretome, and others involved. So, simultaneous integrated analysis of several layers of cancer regulation has the potential to explain cancer biology in a manner that analysis of a single omic cannot ever accomplish. And this concept is known as multi-omics. Multi-omics can be helpful in both forward and reverse translational research. When we're talking about forward translation, basically what we mean is the path from the bench to bedside. Multi-omic analysis can help identifying and deciphering the complexity of involved factors and finding the leading mechanism to be targeted as either a biomarker or a drug target. But not less relevant and really prominent is multi-omic approach in reverse translation. So basically, a reverse translation is from bedside to bench. When, for example, an existing therapy which is already approved and used in patients, it could be potentially improved by identifying a new biomarker or a new possible combination to augment the response. So quite often we see examples of reverse translation in immune oncology, and although immune therapy are highly effective therapies, just a small proportion of patients do achieve significant clinical benefits, and this is why many ongoing studies and clinical trials are focusing on identification of better predictive biomarkers and potential drug targets to augment immune therapy response. And I will give you an example of reverse translational studies later in my talk. But just for now, I just wanted to mention that multi-omic research is becoming increasingly prevalent in biology overall. Currently, we see in 63% increase in publications, including multi-omic studies, and also about 50% year-over-year increase in grants for multi-omic studies. And this is reflective of research across fields, suggesting that combining characterization of multiple layers of cellular regulation enable researchers to observe things that would be hidden with one type of data alone, such as causes underlying differential patient responses to targeted cancer therapies. On top of that, and just to make sure that we are on the same page regarding terminology, here what you can see on x-axis, let's say, there are 4 major omes plotted, like genome, transcriptome, epigenome, and proteome. And here you can see modalities which can be analyzed in these omic combinations. And this chart here shows commercially, with check marks, you can see commercially available comprehensive NGS workflows to analyze samples. And as suggested in the previous slide, Illumina sequencing and microarray technologies offer the flexibility to characterize every ome. So if it is necessary to understand proteomics, epigenomics, genomics, and transcriptomics, to fully characterize cancer cells, all of that can be done through Illumina sequencing platforms. And when performed in a multi-omic manner, the comprehensive picture can ultimately contribute to discovery of biomarkers and drug targets, further enabling precision medicine. So in the next section, I would like to give you a couple of examples how multi-omic studies can really accelerate our understanding cancer and improve therapeutic options. Here's a wonderful study showing the power of multi-omics in immune oncology. As I mentioned a few slides ago, prediction of immune therapy response is highly challenging, as none of the currently existing biomarkers can reliably predict outcome in these patients, though, for instance, PD-L1 staining, TMB, tumor mutational burden, or immune repertoire are one of those currently used biomarkers, just to mention a few. So the authors hypothesized that the multi-omic dimensional approach, modeling both tumor and immune-related molecular mechanisms, would better predict immune checkpoint inhibitors response than simple mutational-focused biomarkers, such as, for example, tumor mutational burden. They did analyze paired pre-treatment FFPE tumor and normal blood samples from patients with late-stage melanoma. And they used whole exome sequencing and RNA sequencing to first analyze tumor mutational burden, T-cell receptor beta clonality, and neoantigen burden. And each of these proposed biomarkers, as you can see here on the right, could differentiate responders from non-responders, but statistical significance, especially in these two cases, was not really high, actually on the edge of statistical significance. As next, in contrast, when the authors used data derived from whole exome sequencing data and RNA sequencing, and they used their proprietary analytical pipeline, which produces comprehensive tumor mutation information, gene expression quantification, neoantigen characterization, HLA typing, T-cell receptor repertoire profiling, and tumor microenvironment profiling. This all was combined into so-called NEOPS score. And when they established this score, they actually could show that, here we go, that actually could show that this NEOPS score, as combination of all of these factors, could highly significant separate responders from non-responders, and here you can see Kaplan-Meier curve demonstrating beneficial progression-free survival for those patients with high NEOPS score. And actually, this is a great study showing that combining genomic and transcriptomic data really improved predictive power for immune checkpoint inhibitor therapy outcomes than one ome alone. So that was an example of combination of genome and transcriptome, but we also know, when referring back to the central dogma of molecular biology, that genomic information is transcribed into transcriptomic information from DNA to RNA. But this process is highly regulated by the epigenome. And when talking about epigenetics, we know that there are of course more, but I would like to mention the three major regulatory mechanisms like DNA methylation, here chromatin accessibility, and also DNA-protein interaction, so involvement of regulatory factors. And just to highlight how combination of genome, transcriptome, and epigenome can accelerate our understanding, accelerate cancer research, I brought this study from Japan. This is a wonderful illustration of multi-omic approach for new drug target discovery. As you might know, gastric cancer has a high incidence rate in Japan, but also globally, it's the third leading cause of cancer-related death. Patients with late-stage gastric cancer and also patients with gastric cancer with malignant ascites, which are about 10% of gastric cancers, they have very limited therapeutic options. So the authors hypothesized that combining whole genome sequencing, RNA sequencing, and epigenetic analysis may stratify gastric cancer patients by specific signatures, identify molecular targets enriched in subtypes, and explain targeted therapy resistance. What they did, they enrolled patients with late-stage gastric cancer and took malignant ascites from these patients here, and then they cultivated tumor cells to establish tumor cell lines from patients. These tumor cell lines were analyzed then for RNA sequencing to see if there are differentiating signatures in these cancers, in these tumors. Then they also did whole genome sequencing to see what genomic signatures are involved and basically underlying transcriptional changes. And they also did methylation profiling and ChIP-seq to infer what epigenomic factors can explain differences between groups and explain differences between transcriptional signatures. So first of all, doing transcriptomic analysis, the authors noted that expression patterns actually clustered all samples into two groups. And when analyzing the difference between the two groups, they saw that groups are differentiated by EMT gene set enrichment. And EMT stands for epithelial to mesenchymal transition. Cancers, by definition, they arise from epithelial tissues. So when they lose epithelial characteristics and gain mesenchymal characteristics, this is always associated with a progression of cancer and metastatic outgrowth, so really unfavorable process in cancer patients. So basically, EMT alone could be already used as predictive biomarker in gastric cancers. To validate this finding, the authors took publicly available data from TCGA, and what they saw that indeed EMT high patients with EMT high signatures, they performed worse in comparison to those without EMT signatures, but unfortunately it was not statistically significant. But actually it was not the major aim of the study, right? Because the authors wanted to see, to analyze the link between the consequences, the presence of two, let's say, EMT and non-EMT groups, and the causes, so potential involvement of genomic and epigenomic levels to find pathways and maybe genes which could be targeted in these patient groups. So to do so, they performed whole genome sequencing analysis, which revealed that MET amplification and CDKN2 and B loss were enriched in EMT group, and these aberrations were present in non-EMT groups. And also analysis of the epigenetic level, here we go, using ChIP sequencing showed that EMT group was dependent on SMART3 super enhancer, whereas a non-EMT group was rather dependent on these super enhancers. And also they did methylation analysis using Illumina EPIC array and revealed aberrantly methylated loci in EMT and non-EMT group, and the most pronounced differences was relatively reduced TAS methylation in the EMT group. So these were the findings. As next, they wanted to see what potential therapeutic targets, therapeutic strategies could be employed in this EMT group. As I mentioned, EMT is a process which is associated with unfavorable outcome or leads to unfavorable outcome in patients. So basically, especially for this group, therapy options are highly needed. So obviously, as MET amplification is present in this group, MET inhibitors could be used, but we know also from literature that MET inhibition in gastric cancer is controversial and so far has failed to prove clinical benefit. And this is why the authors were looking to identify new potentially druggable options. And doing analysis of loss of function, screen in cells, they demonstrated dependency of EMT gastric cancer cell lines on TET, TET pathway rather than on SMAD3. And this was actually great finding because TET can be inhibited by TET inhibitors. And this is what they did. In mouse experiments, they took mice with gastric cancer and treated them with either placebo or K975, which is a TET inhibitor, and they could indeed show that in the treated group, they observed significant tumor suppression and significant improvement of overall survival of these animals. So the authors confirmed the therapeutic potential of inhibition of the TET pathway is in the EMT group is actually present, indicating that this could be a potential way to therapy these patients. Of course, further research is needed. And also we know from the literature that TET pathway is one of the causes of resistance to growth signaling inhibition, and actually co-inhibition of MEK and TET can have synergistic effects, and also combination of TET inhibitors with other agents could be a promising approach to overcome the therapeutic resistance in gastric cancer with EMT as the group with the worst prognosis from gastric cancer. So this is a wonderful example how multi-omic approach can really decipher new mechanisms which can be targeted. All right, the next section, I would like to spend some minutes talking about proteogenomics. Why is it especially interesting? Because traditionally next generation sequencing experiments are used to analyze genome and transcriptome, nucleic acids, right? But actually also protein analysis are possible. And here I would like to highlight just a few methods. So again, starting from the central dogma, we know that DNA is transcribed into RNA and RNA to proteins, but we also know that presence of DNA does not necessarily mean that protein is expressed, and protein expression is not a linear function of mRNA expression, meaning that many groups and many studies evaluate protein and transcriptomic signature just because there is no direct link between transcriptional activity and protein expression, meaning that these legacy factors, legacy techniques like Western blotting, immune cytochemistry, FACS, and CyTOF are used for protein analysis, and transcriptomic analysis is quite often done using RNA-seq. But the disadvantages of this approach are trade-off for sample availability, and also that only consequent and parallel analysis are possible, but you cannot analyze the same sample simultaneously, exactly really the same cells. With introduction such methods like CytSeq, RIPSeq, and BandSeq, now it becomes possible to analyze protein expression with NGS readout and actually enabling simultaneous analysis of protein signatures and transcriptome in one sequencing run. And how is it possible? It's really elegant. I'm sure you've already heard of this technique and maybe even used it. The idea is really simple. You have antibodies against your protein of interest, and these antibodies are labeled with antibody-derived tags, so basically nucleic acid tag, and everything what is tagged by nucleic acids can be sequenced. So obviously you can sequence this barcode and see if antibody bound the protein of interest. And just to speak about the workflows, they're really straightforward. Here I have examples for two workflows. Site-seq, this is a single cell workflow, and band-seq, this is a bulk workflow. And first of all, of course, you prepare your sample. And the only difference between single cell and bulk workflow is that for single cell, you actually do isolation of single cells after you performed mixture with the antibodies against your target of interest. And for the bulk workflow, you actually do not do partitioning of single cells. And then afterwards, you do lyse your samples, prepare libraries, sequence, and analyze data. And just have a look in our methods guide for more information. But as you can see, this really straightforward workflow. Just an example, let me show you how site-seq can be used to better understand the mechanisms involved in therapy response and to identify new drug targets. This is a study from a group in France, used site-seq to analyze immune landscape in patients with chronic lymphocytic leukemia, which is one of the so-called B-cell malignancies. And for CLL, patients usually become ibrutinib, which is standard therapy for this group of malignancies. But of course, at a certain point, resistance becomes a problem. So the group analyzed patient samples before therapy at M0, at response, so M3, and at progression, N27, so 27 months after therapy. Using site-seq, the only method they used in this study. So first of all, I would like to draw your attention to this graph. So basically on this UMAP, two-dimensional UMAP, you can see the merged representation of phenotypic and transcriptomic signatures. As with site-seq, you can infer both phenotypic and transcriptomic signatures. And here you can see this representation showing that, for example, CD3 cells, they could be separated from other cells based on mRNA signatures, as shown here. I hope you can see it, though it's really small, and also based on cell surface proteins. But for example, for CD4 cells, it was not the case. Some mRNA signatures were not sufficient to identify CD4 cells, but only using antibody-derived tags, so basically only based on cell surface proteins, CD4 cells could be identified. This is actually the power of the method, showing that combined analysis really gives you more insights and more analytical power. But this is, let's say, just a technical aspect. And of course, there is much more interesting scientific data behind. So now when looking on B and CLL-like cells population, which appeared as phenotypically and transcriptomically homogeneous, the authors performed also unsupervised principal component analysis and revealed interclonal heterogeneity, as shown here, despite transcriptomic and phenotypic similarity. And also, they did analysis at these three different time points of blood collection and saw the three B leukemic cell clusters segregated according to treatment time. And this ultimately led to identification of B leukemic cell subpopulation exhibiting so-called MC2 signature, which significantly differed from all other CLL cells during ibrutinib treatment. Why it is so important? Just because, well, interestingly, MC2 cells subpopulation was present before treatment, already before treatment, but also MC2 cells were highly enriched during progression, suggesting that these cells could have more aggressive potential. And the fact that this signature was already detectable before treatment and reappeared at later time point, at time point of progression, indicates that it can be utilized as a predictive biomarker. But as next, of course, going deeper to understand biology, MC2 cells demonstrated upregulation of MYC-targeted genes, as shown here. And why is it so important? Just because c-MYC can be targeted. It plays a role in cell cycle progression, as we know, and targeting MYC by BET inhibitors is currently evaluated in clinical trials.
Dr. Babayan?
2 minutes.
Yes, thank you.
Thank you.
So that's actually also interesting study to just highlight the power of the method as just site-seek alone was used. And now in the remaining 2 minutes, I would like to speak about the topic which is really close to my heart, about liquid biopsy and multiomics in liquid biopsy. When we talk about liquid biopsy in blood, we talk about analysis of tumor-derived material, and this can be circulating tumor DNA, circulating tumor cells, proteins, extracellular vesicles, tumor-educated platelets, many different analytes. And actually, multiomic analysis and multi-analyte analysis in liquid biopsy is also the way to accelerate shift towards precision medicine. And just really to finalize my talk, I would like to highlight this wonderful study called ELIMA study from Germany, where the authors analyzed 2 different omes, so genomic and transcriptomic level, using 3 different analytes, so extracellular vesicles, circulating tumor cells, and also cell-free DNA from blood of breast cancer patients. And they could show that each of these analytes and of these analytes alone actually could help to identify patients with targetable with tumor signals, but actually just when combined, then the power of identification of tumors of patients with tumor-derived signal really increased over 95%. And when combined into a score, this demonstrated significant prognostic value, actually showing the power of multiomics and of combined analysis. And with that, I would like actually just to highlight that we couldn't speak about everything. We spoke about bulk and single cell analysis, liquid biopsy, just touched on it, but of course there is also spatial modality, which is really interesting. And here on the right-hand side, you can see some popular omic combinations. I couldn't cover all of them. And I invite you to download the multiomics ebook to read more in depth about multiomic approaches and how it can help you to empower your research. And with that, thank you very much for your attention.
Thank you very much, Dr. Babayan. Unfortunately, we only have time for one question from the audience. Yes, please, microphone here.
So thank you very much for the talk. Very interesting, the proteomic approach using Illumina, very innovative. But how do you envision its applicability with clinical samples? I mean, I buy the liquid biopsy thing, but the paraffin-embedded samples, do you think there's any protocols being adapted to be able to perform studies on proteomics by NGS?
To my knowledge, yes, you can adapt some workflows. For paraffin-embedded samples, what is especially interesting is, for instance, spatial analysis. I know it's on proteomic, but it gives you a completely different resolution, right? Because you can see transcriptomic signatures in spatial context. And since you have spatial context, you can also do stainings, right? Immune cytochemistry stainings to infer proteomic makeup. At the moment, I think it's not possible to do it on one slide, but if you use just 2 slides from the same, 2 consequent cuts, that probably gives you the full picture.
Okay, with that, we thank you very much, Dr. Babayan, and hope to see you soon in Spain. Okay, for the rest, we will have a coffee break, so please, you can find coffee outside. Be careful, maintain all the COVID-related regulations while you are enjoying your coffee. And for the attendees, virtual attendees, we will be back in 25 minutes. Thank you.